Thanks very much, Peter. Very, very kind introduction. I would really like to sincerely thank the organizers for putting together this meeting. I was thinking last night, I mean, it's been about 12 years since we met the first time. It was just a handful of investigators. I mean, I see most of them in the audience. And then over these years, like this whole field of MPS, of microphysiological systems or organs on a chip, exploded. And this is really telling a lot about the science, about engineering, about our ability to work effectively with each other. And also it's interesting, I mean, I was some 25 years ago, I was very fortunate to be part of an emerging field of tissue engineering. And then this is like another revolution in our field. Like 10 years ago, 12 years ago, we started to work on these tiny technologies on microsystems. So it's really a pleasure to be in this meeting, and the quality of science is just totally amazing. So I'll talk today about specifically about organs on chip that involve multiple tissues and that are designed to model some of the systemic diseases. This is how we started to think about it, and then we started to realize that actually most, if not all, of our diseases have this systemic dimension. So bringing in immune component, bringing in vascular circulation is always very, very helpful to get a system of higher biological fidelity. So just to mention a few disclosures, they really don't affect anything that I'm going to say, but I just feel obliged to do this. So going maybe starting from tissue engineering, what it is and where is it going. Pretty much every recipe for tissue engineering has these three components that you see here on this cartoon. They're the cells, which are the most important and central component of every tissue engineering system, because there is no other way to make or maintain tissue but by living cells. So I'll talk today exclusively about human stem cells as a cell source for the tissues that we are studying. We are instructing the cells about what to do, because stem cell can do pretty much anything. We're instructing them by these two bioengineering components. One is the scaffolding material, which is designed to be tissue specific and to mimic the extracellular matrix by its molecular composition, architecture, and mechanical properties. And the other is a culture environment or a bioreactor, which is a kind of substitute body providing environmental control, supply of nutrients, removal of metabolites, and very importantly, provision of regulatory factors. So you have this triad that has been driving the field of tissue engineering towards clinical applications. So for many years, tissue engineering was fully dominated by working on a clinical scale and fixing bones and hearts and lungs and many other tissue and organ systems. And this will continue. Not much will change in this area. However, some 10 years ago, it turned out that the knowledge gained and the methods developed and resources developed for clinical scale tissue engineering can be scaled down to making millimeter-sized tissues and connecting them into physiologically meaningful units, so that we don't necessarily do tissue repair organs or tissues that are lost to injury or disease, but we can use these units for modeling of development, of regeneration, and of different diseases. So this is what we are talking about today. 10 years ago, the federal agencies play a major role in really launching this field by posing a question to the scientific and clinical community. Can we utilize the knowledge that we have gained over the years in the field of tissue engineering and stem cell biology to make models of human tissues that will help us bridge from discovery into the clinical studies. So this transition has been made over many years by using either simple simple human cell cultures or animal models. Now the question was, could we make minimally functional units in form of human tissues and then use these units to study first toxicity, adverse effects, and then also efficacy of some of the drugs. So the whole effort was very, very much oriented towards drug development, and the reason is very clear. Bringing drug to the market is one of the least reliable and most expensive processes that we have today. So the question was, can we do better? What turned out to be the case is that actually this effort of serving the drug development had much broader impact than originally envisioned. And actually the MPS turned into an effort of modeling human physiology across different biological scales in a way that is serving both the science and many, many branches of medicine, not only drug development, not only regenerative medicine, but also our understanding and treatment of different kinds of diseases. The MPS also brought to us this need to work in a patient-specific context whenever meaningful and necessary. And actually what you see here is just a schematic, which is a very a partial picture of what we are doing, meaning that there are cell sources such as induced pluripotent stem cells that we today routinely develop from blood. You just need a small test tube of blood. And these cells can give rise to many, many, many different cell lineages. And we are talking here about the tissue-specific cells, such as hepatocytes for engineering liver, but also supporting cells that are absolutely necessary to make functional tissues of different kinds. So it is really wonderful that we can today start from iPS cells and then make different types of tissues that are all derived from the same individual. So then you can make these physiological units, sometimes just a single tissue, multiple cell types, they're always multicellular, sometimes multiple tissues linked to each other by vascular perfusion or possibly innervation that are all isogenic, they're all genetically clean. And then you can imagine that this also opens door to studies of biological diversity. We are all different from each other, and introducing sex and age and ethnicity and state of the health or disease as an experimental variable is hugely important. So following this paradigm, we can make many different tissue types. Here is just a small subset to illustrate the diversity of these tissues. So you can make a muscle, you can make bone, you can expand this bone to include bone marrow, you can study barrier functions of endothelium and epithelium, etc. And you can also make some additional supporting cells such as immune cells. Today we are able to derive many types of immune cells also starting from iPS. So patient specificity and the broad diversity of the tissue types that we can make. So let me just show you two examples of tissues that are very different from each other, bone marrow and heart muscle, to illustrate what is it that we can do in terms of engineering and maturing these tissues, and also to show utility of these tissues even if they're used individually. So when we make bone, we are starting from iPS cells that can be a source of mesenchymal cells that you need to give rise to osteoblasts. So this is the osteogenic differentiation. But then you also need to make a bone that is really realistic biologically. You also need to include osteoclasts. So osteoblasts are building bone, osteoclasts are degrading bone. So bone is really healthy and functional if these two processes are happening together. Osteoclasts have a different source. They also come from blood, but they come directly from monocytes. So this is the bone that we make, and we have shown over the years that actually inclusion of both of these components is very, very helpful to get more realistic molecular makeup, ultrastructure, and mechanical properties of the engineered bone. So this is the old work. Much more recently, we started to populate this bone niche with another type of cells, which are hematopoietic cells, CD34 positive cells. We started by just getting primary cells from the body, and then about a year ago, we started to deriving these cells again from IPS for reasons that I explained to you. After several weeks of cultivation, we can show that this bone marrow benchmarks very favorably against its native counterpart, normal, healthy human bone marrow. You see some stains here. These are the stains that you are supposed to see. You see also GAMSA-right stain for the blood cells that this bone marrow is generating. And at the right, there are a couple features that are really very important to show that this bone marrow has utility. One is the colony forming ability. This is a standard assay. And the other is what's labeled here as hematopoietic expansion. So you want to see generation of CD45 and CD34 positive cells. And you see here that this is over a period of a couple weeks. So the expansion is about 100-fold for these two cell types. This bone marrow can be used in two important ways. One is that we are plugging it into our multi-tissue platforms. I'll show you examples to serve as a source of macrophages that are very, very important, both resident and circulating macrophages for the functionality of many tissues. But this bone marrow can also be used as a model of leukemia. So this is where we are collaborating with clinicians who are providing us with the patient's primary leukemic cells, and we are helping them to model the progression of disease and to test some of the treatment modalities. So this was tissue number one. Tissue number two is the heart muscle. So we've been making a heart muscle for many years now, again starting from iPS cells. You derive cardiomyocytes, you derive supporting cells such as cardiac fibroblasts and endothelial cells, and more recently resident macrophages. We encapsulate these cells into hydrogel that is designed to mimic the heart matrix. And what we do in culture is after the muscle assembles, it takes about up to 1 week. Then we start stimulating this muscle, conditioning it by exercising it. Essentially, by electrical signals, we make the muscle twitch, the same mechanism that our heart is using to pump blood, to twitch and pump blood. And actually, this stimulation is being applied at an increasing intensity. So we exercise the muscle, and this is what makes the muscle stronger. For this to happen, it was very important to find out that we need immature cardiomyocytes, which was completely opposite to what we were doing for many years in the field, always thinking like more mature cells are better. Yes, they are, but actually if you are developing a tissue, then immature cells are better responding to stimulatory factors. And what you see here at the left is the cross-section with cell bodies and membranes, and then you see this very thin lines. So these are these invaginations called transverse tubules that are enhancing calcium trafficking. And on the right, you see very nice ultrastructure with orderly registers of sarcomeres and a lot of mitochondria that are actually present at physiological density. Metabolism is aerobic, so this is postnatal metabolism, and electrophysiology is also mimicking many aspects of adult human physiology. What do we do with this muscle? We obviously use it to study toxicity because cardiac toxicity is responsible for about 1/3 of all cases of toxicity that is observed clinically. But we also use this heart muscle to study different diseases, genetic diseases, and also injury. And the example that I show you here is a model that we recently established. This is still work in progress, still not published. It's a model of ischemia reperfusion injury. In simple English, this is what happens following myocardial infarction. And because we are able to keep this muscle in culture for a long time, we can study both acute and prolonged or chronic effects. So there is a protocol, this is schematically shown here, that allows us to establish conditions that are similar to those in the infarcted heart, so in the muscle that is deprived of blood supply. And then we go after 6 hours of this ischemia, which is not only lack of oxygen, but also changes in potassium, changes in lactate, in pH, and whatnot. We follow up with reperfusion. Both of these conditions cause injury to the muscle, and we are modeling this injury. And you see here at the bottom left that actually many of the parameters from viability to active force go to about 50%. This is what we determined. We actually developed this protocol pretty empirically to result in this 50%, which is what you see clinically. What is very nice is this image that you see here, histology, shows you that we are also able to see this deposition of very large patches of collagen, which is this fibrotic process that happens in the heart following infarction. We also included into these studies sex-specific studies, so we are looking into differences between males and females that are known to exist. And this actually turned out to be major effort because we were discovering that many of our culture media contain hormone-like substances, so we had to develop new culture media that are hormonally neutral so that we can introduce the hormones. So it's not only genetics, male, female, it is also the presence of the right hormones that we are now studying. So these were the individual tissues. So let me now talk a little bit about the combining these tissues into physiological units. This image is actually from this 12 years ago when we started to respond to the requests of NIH and other funders to develop something that pharma can use. And you can see here one schematic of what we are trying to do at that time, which is to start from iPS cells to derive all these lineages that I showed you, to form tissues, to place tissues into these platforms, and then connect them with vascular perfusion. So we were trying to mimic the human physiology in this way. At the right, you see the design requirements that we actually really declared that we were trying to meet, and this was in response to the requests of our NIH sponsors and pharma. First, it was very important to make these platforms modular and configurable, so you can connect, you can choose the tissues that you're interested in, you can connect them in any numbers and any order. I mention numbers because if you need to achieve, for example, certain level of liver metabolism, and your module with this millimeter-sized liver doesn't give you enough, you can connect 2 livers or 3 livers and have the ratio of metabolism to unit heart tissue or bone tissue or any other tissue at the level that you like. Order is also something that you change. Biological specificity is very important, so this is what I talked about. This is taken care of by the use of iPS cells to a large extent. Now, the very difficult requirement, the non-negotiable requirement was that we need to establish and then maintain mature enough tissues for a period of not shorter than 4 weeks. So this is what pharma was requesting. But we also wanted to connect the tissues, to have this crosstalk, to let them talk to each other. And these are the conflicting requirements, because as soon as you let culture media mix with each other, you may start losing the tissue phenotypes that you originally established. So stable and mature tissue phenotypes for long enough time. So we started with a month, now we are at the several months, and some anecdotal data that we can keep tissues in culture for 6 months, and in one instance even 9 months, along with the tissue connectivity. And then finally, an engineering requirement in order to do dynamic studies, longitudinal studies, to introduce disturbance and see how the tissues are responding, you need as many functional readouts online that are done non-invasively as possible. So this is our wish list. How do we do that? I'll just comment on this difficult requirement, which is to reconcile the stability of tissue phenotype with the need for tissues to communicate to each other. So whenever in doubt, we just look into how does the body do it. So this is really the biologically inspired design. And then what you see at the left is just a schematic that shows us that bone and heart and liver and any other organ you can name it, they have their own environment. Our body is based on compartments and boundaries. And we are trying to make the compartments and boundaries in our platforms. And then how these compartments talk to each other, they talk by vascular perfusion. Between the intravascular space and intratissue space, there is an incredibly important boundary, which is endothelium. And endothelium is selectively permeable. So molecules, extracellular vesicles secreted by cells, and some of the cells themselves, such as immune cells, can cross this barrier. So this is the conceptual design. So this really nice drawing is from the paper that Lucy Lowe and Dan Tagel and colleagues have published a couple of years ago. So this is how it looks. This is the size of like USB chip or microscope slide. So you have different, in this case, 4 tissues. This is the work that we published that I'll comment on a little bit more: liver, heart, bone, and skin. When you look from the top, you see these different tissues. These different colors just indicate that in each chamber there is a different medium. There is a liver-specific medium, bone-specific medium, etc. When you look from the bottom, all you see is endothelium, because the tissue compartments are separated by vascular circulation, which is underneath by endothelium. So we put together these platforms as shown in this little video here at the left. You assemble the platform, you place barriers first, endothelial barriers, and then you go with the tissues that you place into their compartments, each tissue in its culture medium, and then you start perfusion, and then we normally introduce additional cells into the perfusion. I already mentioned macrophages, and I'll also mention cancer cells for some of the models that we are studying. It's very important to work with mature tissues, because our plan as a field is to be predictive of adult human physiology. So I mentioned for the heart that we are maturing the heart muscle. We are forming it from very immature cardiomyocytes. So these are nascent cells that are committed but just start beating, contracting. This is around day 11 or 12. And then we subject it to this regime of like very strict exercise. What we found is, you see here at the bottom left, if you look into the response of this heart muscle to isoproterenol, to increasing concentrations of isoproterenol, what you would need to see is are the responses in terms of measured force that we do by looking into pillar deflections and calcium in green. So this is the response of normal adult primary human cardiomyocytes. What you see in black are immature iPS-derived cardiomyocytes. So if you don't do maturation, if you mature the tissues, then the responses are in red, so they get pretty close to what you see here in green. So what we do with these multi-tissue platforms is we assemble them from mature tissues. Because different tissues take different time and conditions to be matured, we do this individually. Interestingly, bone takes the longest. We need 6 weeks to get relatively mature bone. We need 1 month to get relatively mature heart muscle. So we start at different time points so everyone gets to the finish line at the same time, and this is when we assemble the platform. This is how it looks for this particular case. So you see these 4 tissues here, 1, 2, 3, 4, and this 5th compartment is for the vascular, for the endothelial cell medium, which is our perfusate. As I mentioned, the endothelium is selectively permeable. This is very important. This is how the endothelium looks. It's very interesting that we form endothelium by coating very porous and elastic membrane by endothelial cells. We let them attach. We add supporting mesenchymal cells or pericytes. They need some support. And then we condition this endothelium to become confluent and quiescent by ramping up the shear. This is like a known protocol in the field. While doing that, some of the endothelial are underneath liver, the other are underneath bone, underneath heart. We see differences in endothelium adjacent to different compartments, with the liver compartment characterized by the leakiest endothelium, which is also the, as you know, the feature of the normal liver tissues. So this endothelium is able to let the macrophages pass through the endothelium. Interestingly, the macrophages and monocytes, they just go by all healthy tissues. Here you see heart muscle, which is healthy, nothing happens. The monocytes were labeled green, so you see no green in this case. But if we do injury to the heart muscle, monocytes find a way to open endothelium, to get across endothelium and invade tissue. And this is this green that you see, so this is heart tissue invaded by immune cells. And what you see at the right is also demonstration that extracellular vesicles or exosomes are crossing endothelium. What we have done here is we gene edited iPS cells that we use to make the heart muscle. So we introduced CD63 that was linked to GFP, so we can see cells are green and exosomes also are green. And these exosomes originated from the heart, but then we found them in all other tissues. So they are really getting around, and they are a very important communication vehicles, as we know in biology. So the obvious question was, it looks really neat and it mimics the in vivo situation. Is it really important to have endothelial barrier? So this study was really designed to answer this question, and based on this answer, to do PK/PD studies of a drug to show utility of the platform. So we studied here the platform that I already showed you with these 4 very, very different tissues, liver, heart, bone, and skin. And then there is endothelial barrier underneath. And we studied this platform in 2 variations. One was what I just described to you, where each compartment, you have these compartments, you have recirculating flow, and elastic membrane, porous elastic membrane shown in black, coated by endothelium from underneath. So this is what we call the interorgan. They're like connected to each other. The other variation of the platform was everything the same but no endothelium. So we wanted to compare these 2 and see what is endothelium doing. And we also wanted to see how each of these tissues, for example, liver, performs when in contact with all these other tissues compared to liver alone under optimal conditions, optimal culture medium. So there are these 3 groups. So the group with endothelial barrier and circulation, no endothelial barrier with circulation, and then the individual compartments. So this is how just another view at this. So this is our interorgan platform with endothelium. This is our mixed platform with just a membrane, so elastic membrane without endothelial cells. And then we have these isolated tissues that we cultured both with and without endothelium and flow. And what we have done in this study was to look into the toxicity of doxorubicin. Doxorubicin is one of the very common anti-cancer drugs. The use of it is an effective drug, but actually its use is limited by toxic effects that are on the heart tissue and liver tissue in particular. So we have done these PK/PD studies with all configurations of the platform. So there is the entire platform with and without endothelium, and then single tissue with and without endothelium. What we have found in this study is that in this platform with endothelial barrier, we were able to maintain the tissue phenotype shown here just by histomorphology at the level very comparable to that for tissues in isolation, which was not the case for the mixed platform without this separation. We looked into a lot of assays. So one is shown here. So this is a GO analysis where we looked into proteins that are shared, that are expressed in more than one compartment. For example, if you look here at the left for interorgan platform, we looked into proteins shared between liver compartment and heart compartment, liver and skin, liver and bone, etc. And then proteins that are shared across all these tissues. So we found 14 out of very, very many. And interestingly, these 14 proteins are a sort of common that you really want to see that are related to cell well-being and normal function. In the mixed platform, this number is 40. And when you look in more detail into what these proteins are, many of them are the ones that you don't want to see. You don't want to see liver protein in bone, etc. And then in the isolated culture, it was in between. And this tells us that isolation is positive in a way that you don't compromise the tissue phenotype, but actually some of the tissues, in particular bone, were a sort of missing the communication to develop to the full extent. We also looked into how the tissues compare to normal adult tissues. And so what you see here is the shared gene expression. Heart is doing remarkably well. So it's at the level above 92%, like skin is above 80, etc. And then here is a bone that appears not to be doing well, but actually we were very surprised in all these cell and tissue atlases that were published over the last few years, there is no bone. So we just use marrow as something that's the closet. So I'm not sure how realistic is this number here. And then here is just a map between adult is the first column, so normal adult. The next column is our interorgan platform, and then you see mixed, and then you see isolated, and you see that overall mixed is doing the worst in this particular constellation of tissues. We also in this study looked into if we can identify some of the biomarkers of doxorubicin toxicity, and we found one in the heart muscle that was present both in 4 tissue configuration and isolated tissues, as well as in the perfusate. So it was MIR1, which is like a precursor of a whole family of MIRs that are important in this context. So let me now just show you a couple examples in the remaining time. So from this point on, it's all new data. I was thinking it would be nice to show something that's still work in progress, you know, in addition to some published stuff that I showed you. So one application of the platform was to look into the effects of radiation on human tissues. This was very directly initiated by the interest of NASA. They wanted to model how astronauts would be responding to long-term exposure during long flight space missions to the space radiation. The other obvious application is for clinical radiation for treatment of patients. So what we have done here is we selected a subset of tissues that are relevant for radiation studies. One is liver, because we were also looking into protection of the cells and tissues, so the use of different drugs. So we included liver because drug metabolism is very important. We included the heart muscle, which is a known site of delayed, like a chronic radiation damage, bone marrow, which is the site of acute radiation damage, and then we have endothelium, which is needed to establish connections, and we also included immune cells, which are generally important and are also important for the response of tissues to radiation damage. So just a snapshot of what we found here. Very simple platform. So we have heart, liver, and bone marrow, and then endothelium is present here. Circulating cells are present here. And so what we have done is the control, so not irradiated, nothing bad happened to this platform. And then we had different types of radiation, neutron and proton, and we had different levels of radiation. So what you see here is how, at the left, how the bone marrow responded. So there are two populations of cells that are important, CD45 positive and CD34 positive. What you see here, this is the baseline, how many cells we have, CD45 cells, being generated by bone marrow. So this is the baseline, the first bar. The second bar is exposure to photon radiation. And then you have exposure to radiation, but of the tissue, bone marrow tissue, that was supplemented by GCSF, which is a radiation protection agent. And you see that actually much of the effect is avoided. And then another type of radiation also with the GCSF. A very similar situation for CD34 positive cells. For the heart, qualitatively similar. What we measured here is a function, excitation threshold, how easy it is to stimulate heart to beat, and the maximum beating frequency, which means how responsive the heart is to electrical signals. And you see that actually both of these properties worsen. So excitation threshold goes up, which is bad, and maximum beating frequency goes down, which is also bad, in response to radiation, but actually much of the effect is avoided by a radiation protection agent. So this is really just a proof of principle that these kinds of things can be studied. So now I'll tell you in a little bit more detail about a model that we've been developing over the last few years, which is the cancer metastasis, like a cancer patient on a chip type of situation, where we are trying to model some of the key events in spreading of the primary tumor cells into the metastatic sites in the body. So the cells need to shed from the primary tumor. They undergo what's known as intravasation, so they get into the vascular circulation. Then they activate bone marrow, so there is a recruitment of bone marrow-derived cells, very important. And these processes are a sort of priming the metastatic niches so that the cells, like the last step is that these cells are extravasating, getting out of the vasculature and into the tissues. Very, very complex process. And what we try to do in this project is to answer two questions. One is, can we recapitulate metastasis per se? Can we show that the circulating cancer cells are finding these target tissues and colonizing them? And the second question, so like for a specific patient, how likely it is that this patient will develop metastasis? Obviously, very important question because most cancer patients suffer, I mean, the most of the death toll is due to metastasis, not to the primary tumor. So this was the first question. Can we recapitulate metastasis? The second question, if so, can we tell which organ is likely to be affected? Is it going to the brain or bone or liver or somewhere else? So this was the thinking about the experiment. Here are the key components. So we need a vascular barrier. This is the obligatory part of our platform. We included the heart muscle as a negative control. Heart is very, very, very rarely invaded by cancer cells for reasons that we don't fully understand. Is it because there are no stem cells in the heart or something else? But this is a really good negative control. And we looked into two tissues among many that can be affected by cancer cells, bone and lung. I didn't mention, but actually the cancer that we studied was breast cancer, triple negative breast cancer. So these are the among the common metastatic sites. And the work that I'll show you now was done with the lines of cells that were developed by John Massaguet, and he was kind enough to make them available to us. So he developed a line of triple negative breast cancer cells. This is the well-known MDA line that was broadly used, very well characterized. He has done something else which is very important in a mouse. He was introducing MDA cells into the mouse, and then some of these cells would go into bone. They're universally metastatic. Some invade bone and some invade lung. He would then isolate these bone metastasis cells and put them into the second mouse. And after several cycles of this kind, he gets a population of cells that is BOM 1833 that are bone metastatic cells. So when you put them in animal, they only go into bone. And the same story with the lungs. So we had this universally metastatic line, and then we had these two like tissue or organ specific lines. We made a platform just for these two tissues. So we have lung, which was, it's a tissue that requires air-liquid interface. So this is how we matured it. And we had bone, and we had vascular circulation. And then in this vascular circulation, we had circulating cancer cells. And then we did like all analytics that was possible. As I mentioned, for the heart muscle, this is universally true for other tissues. So we matured all these tissues, both tissues and endothelium, before assembling platform together. And this took us about 6 weeks for bone and a little bit shorter for the lung. And then the integration study, the study of actually the metastasis, this first study was done over the time of 2 weeks. I showed you before the bone formation. Let me just show you lung a little bit. So we see here that you have like some very nice immunostains that show you some of the key markers of the lung tissue. And then you have two data graphs here. One is that there is a very stable population of goblet cells that are very important for the function of the lung. And here you see that there is a very big jump in the resistance that we measured by TEER, so this transepithelial resistance of the lung. So the barrier function was established. So the lung epithelium developed this very tight barrier. And so we first introduced MDA cells into the platform. And what you see here in these three images is the view from underneath. So you see endothelium in blue, and MDA cells were labeled green. And the left is heart, nothing happens. At the lung module, the cells are starting to go to the endothelium. So this is the first phase. Later, they will go through the endothelium and into the tissue, and the same for bone. So these two tissues are being invaded, and the heart tissue is not. When we take a closer look at these two tissues, what you see here is endothelium in red and cancer cells in green. The first row is bone, the second row is lung. You see that over just 2 weeks, these very, very aggressive metastatic cells actually expanded, they proliferated, they really grew in numbers very significantly. So we do show possibility of recapitulating metastasis in this model. So the time profiles of the cell migration, MDA cell migration, are shown here. So you see that we started with a relatively small number of cells, and then this is a logarithmic scale. So we go up by 2 orders of magnitude in the cell number, which is consistent with the images that you have seen. So we were looking into the presence of cells by following the markers. So you can also see here, day 1, day 7, day 14. For the 2 tissues, which are lung and bone, you see the increased presence of the cells that you can see visually. And it's nice that this is one of these longitudinal non-invasive measurements, so you can just measure over and over time. Now the second question was about organ tropism. So this here is an experiment with the bone metastatic cells, and we offer them both bone and lung, and you see that they very preferentially go into the lung over this period of time. And the same is for lung. Lung cells go, bone cells go into bone, lung cells go into lung. Tissues that are being invaded change. So you see here the bone metastasis. In the top row is normal bone that hasn't seen the cancer cells. In the second row is the bone that has been invaded with cancer cells. You see this increased presence of TRAP, and then like the areas where you see very high density of these cells. Very interestingly and unexpectedly, we were able, in this case, and also with prostate cancer lineages, another project, to show that we can obtain both the osteoblastic and the osteolytic lesions that are seen clinically. And a little bit more granularity, just to show you that actually metastatic sites are being remodeled, and maybe just to pick a couple examples, like as a result of MDA cell invasion, the presence of osteopontin increases, which has been associated with pre-metastatic conditioning. You see that, for example, osteocalcin goes down very dramatically, calcium release goes up, etc. And then you see like the similar situation for the lung. 2 mucin markers are shown. They both change with the presence of these invading cells. And then just to like briefly recap this graph, the effects of bone metastatic cells on bone remodeling were much more dramatic than the effects of MDA cells. Just briefly, where we are going now, we are working on developing an immune chip that can be used for a number of different applications, including infections. We are also trying, I mentioned at the beginning, to extend the duration of culture to 6 months. We teamed up with a colleague from Baylor so that we can, at Columbia, we are culturing bone marrow and the heart muscle linked together. She is culturing bone marrow and brain organoids linked together. So we are looking into longevity and ability of these tissues to be models of ischemia, but we are using the very same platform, same methodology, same everything. So this is an effort to do cross-validation of some of the data between the 2 labs. Are we both seeing the same effects, for example, on bone marrow? This is how platform looks. I wanted to show you these photos that are very pretty, but they show you an artisan device. This is what we make for research. This tubing here goes into peristaltic pump. I mean, each incubator is a rat nest. It requires a lot of training to use this platform, and this is not something that you can start sending out to your colleagues and sharing data and experiences. So we started to work on turning this into a user-friendly a platform, which is really push button. So it has an onboard micro pump. it is this stack of 6 platforms that you see here actually came from a robotic device that is manipulating these platforms, placing them into imaging devices, liquid handlers, etc. So these kinds of hardware is now getting ready. We already started to share with some of the collaborating labs, but we are planning to share it much more broadly. And now, in closing, just 2 things to say. One is, in addition to science that I illustrated, described to some extent, our lab is a home of the NIH Tissue Engineering Research Center. So our job is to make tools, tissue engineering tools, scaffolding materials, bioreactors, organs on chip, imaging modalities, codes, and whatnot. We have a lot of things that we can make available. So if you're interested, here is the website. You let us know. There are tissue talks that are still going strong every Wednesday, 3 o'clock in the afternoon, actually 9 o'clock this local time, where we invite experts in the field to give interesting talks about tissue engineering. We have Science Simplified, where students and postdocs are sort of translating important publications into language understandable by, say, middle school student. We have Career Conversations, where we are inviting interesting people to tell us how they got where they are, and this is all tailored to help junior investigators with their career choices. And just a few thoughts at the end. I would like to recap what I tried to tell you today and also to comment on some challenges that we are facing. One very important thing is it's all about convergence of disciplines. It is really, things started to happen when we connected biology, cell biology, molecular biology with engineering disciplines. Our job here, like in tissue engineering in general, in MPS field, is to design environments so that we enable the cells to do their job. And I always believe that biology is the difficult part of it and engineering is easy. If you know what you need to do, you will find a way to do it. Individualized studies and treatments are very important. I try to emphasize this because this is important biologically to understand the effects of all these variables listed here on outcomes, but also for the patient care. And in some cases, like for cancer progression and cancer treatment, it's more important than for many other conditions. So field is like really in a very good shape. It's absolutely fantastic to see everyone here and to see great posters and great developments. But I think we are still facing lots of challenges. Some of them were mentioned also by other speakers. One is rigorous benchmarking and validation. We very much rely on benchmarking against clinical data, but I totally agree with our speaker last night, with Donna, that actually animal MPS systems may be really, really good way to rigorously validate these platforms and to build confidence that they can do good job with predicting human data. As we generate more and more and more data, integration and interpretation of these large volumes of data of different kinds becomes another challenge. And there are all these methodologies that are based on different approaches, including machine learning, that are here to help us. Another challenge is standardization of the platforms, of the hardware, but also standardization of our, at least to some extent, self-sources, methodologies, benchmarks, etc. This is something that can help us compare things across different places and also disseminate or democratize, as some people say, the tools that we have. And to end up on an optimistic note, I mean, we live in the era of collaboration. I think collaboration is the single most important gradient of everything we do, and this is where I hope that it will be continue to be a main driving force of the effort. And a thank you note to my lab. that's incredible, like a really, really fantastic group of people, and our funding sources and our collaborators. Again, we wouldn't come very far anywhere. I mean, whatever we have accomplished would not be accomplished with our collaborators. And thank you so much, and thanks for this invitation. It's a pleasure to be here.